Hi everyone and welcome to the latest in our series of Q&A videos. Today I'm joined by Dr. Andrew Shaw, who's an Associate Professor of Physical Chemistry here at the University of Exeter, and he's also the founder of an Exeter spin-out company, Atomarker. And some of Andrew's research has recently led to the development of a new antibody test for COVID-19. And this was a project that was actually funded thanks to donations from Exeter alumni and supporters. So he's going to tell us a little bit more about it. Thanks very much for joining us, Andrew.
Good morning, thank you.
Now, can I start with a relatively simple question, I guess? What does the test actually do?
So there are two tests that have become important in the antibody, in the response to COVID-19. Now we're all amateur virologists. I think we want to understand there's a sort of PCR test that's a scrape from the back of your nose.
Yeah.
The other test is a measuring of the way we respond to the virus, and that's generated an immune response from their antibodies. So the virus has a number of component targets. So if you look at the virus, it has all these spikes on it that gives it its name, coronavirus.
Yeah.
There are two types of spikes, and we can measure the antibody response to the two types of spike. And also there's something called a nucleocapsid inside it. So our response to coronavirus is a triple response. We can either do S1, spike 2, or the nucleocapsid. And then the body produces an emergency response, which is a so-called IgM response, a first responder, and then that matures into a second phase response, an IgG response. So what the existing tests on the market have done is looked at one or two of those, and our test looks at all of them. So we can see the host response profile, the triple response to the spike proteins and to the nucleocapsid protein, and we can see the early stage response and the late stage response. So we think that's a new type of test and very different from one that's on the market at the moment.
Brilliant. And why is it so useful to be able to know, or to be able to detect the presence of antibodies, but also to be able to do it at this kind of level?
So the sensitivity comes from being able to say it's a triple response. So can I see the patient doing one or other or all of these? Yeah. About 40% of people respond to all three to give a true triple response. Some people don't respond to one of them, so some of the market tests out there will be missing up to 4% of people. So by looking at the triple response, I can catch everybody, and I can then look at how the response evolves. So imagine you're producing a vaccine; they're all targeted at the spike proteins, and we'll be able to see the response to the vaccine, and they're not going to be able to tell the difference between a vaccine response and a host response, say to the nucleocapsid. So we can capture the vaccine response as well as the host response. So we can see all of those. And because we can see the types of antibody response, the early responder as well as the late responder, you can say a bit about when the test is going to be useful. And we've got really good sensitivity at 14 days, and I'm hoping we might push that into the first week of an infection and begin to rival the RNA test with some sensitivity for population screening.
Brilliant. Now you mentioned vaccines there. I mean, what does this test mean for future COVID treatments, do you think?
The big problem now with the flu season coming up is people have had COVID, and there are about 400,000 of those who've left hospital. So that's the minimum number. They are in a strange immune state, a so-called hyperimmune state. And if they get a second dose of COVID afterwards, we don't know what happens. Now, the model is dengue fever. If you get dengue fever once, you survive it. You get it the second time round, the mortality rate's really high.
Okay.
So the big question is whether you get COVID the second time round, is that seriously bad for you? So in a second response, a second wave, that could be a challenge. But what if you now get the flu on top of COVID? Imagine your lungs are about 50 square meters, about half the size of a tennis court. And imagine they were inflamed just like you'd scrape your knee. You can see how your body's worked hard to get rid of that inflammation. It's going to be very red. And if you get a flu infection on top of that, that's going to be a real challenge. So actually, I think the flu season looming is an unknown for the COVID sufferers. So we need to know whether you've had it, and the antibody will tell you that. And actually, as of yesterday in the lab, we got the first result of the flu chip. So now we can see whether you've had COVID and have got the flu vaccine.
Okay.
And I think that could become very exciting into the flu epidemic, flu season as it opens up this winter.
What are the next stages of your research? I mean, you've mentioned flu there. What are the next steps?
The big questions are, if you've had it once, can you get it again? So do you have immunity? And that's the only way we'll solve that is to look at some people that have left hospital. We'll know what their antibody profiles are. And I mentioned the first responder, IgM. Now that doesn't happen if you have immunity. So if you see it again, what happens is you just get the good solid IgG response. So there's a difference in the response between your first time response and a second time response. And if we can follow those people longitudinally over time, we might work out whether people have got immunity. And that's a big question. We don't know that as a society. We don't know that. We need to know. So we're putting in a proposal to set about a big longitudinal study. And I think that would be very important. And then the whole idea of this group of individuals in society that have had COVID-19 and what that does to these so-called comorbidities. Are they vulnerable to the flu? Should you give them a vaccine even? Is a very interesting question. Should you give them the flu vaccine? Are they going to respond really badly to that? So we've got a whole stratum of people that now need to be treated differently in hospital. I think we need to understand that. And we're beginning to talk to government about that too now.
Yeah. I mean, I know this is a slightly different area, but do you think that there will be a vaccine that will be effective against coronavirus based on what you know? Or?
The best guess at the moment is it's a long way away. It could be next summer.
Right.
There are about 50 vaccine projects out there that are all trying. Some of them with the novel vaccine approaches like the Oxford lot group. There are some other more traditional ones like the flu vaccine approach. Nobody really knows, and these vaccines are still quite a long way away.
Okay, cool. And there's one final question, a slightly different angle. Now, this project was funded by alumni to our emergency appeal, which is quite a different way to fund research compared to normal. I mean, what has that philanthropy meant for you?
Speed. It was really good. I chatted to Jonathan and Snicker about it one week. We put the appeal out, put the appeal together, and money started arriving really quickly. So from former students, was quite nice. Undergraduates I taught were donating 10 or 20 pounds or something. But very quickly we got to 50,000 pounds. That meant I could make the chips in reasonable volume. And then we set off some testing at St Thomas's Hospital. So what was a casual conversation turned into some real money. And then a champagne moment in science. We made the chips work. We were there. We took some blood from a sick patient and it worked first time. And we were able to see this evolving host response. You think, good Lord, we can see something here. And then the ongoing donations, it's up to about 140,000 now. That means I've been able to make more chips. We were able to make more tests. We've got to the point where we know it works.
Yeah.
We've even submitted to the MHRA, so we're allowed to sell it now. We have a partnership with Smiths Industries Living, so I can make hundreds of these devices and potentially do hundreds of thousands of tests a week. So what was an initial project with a PhD student behind the face mask in London at Thomas's Hospital, and has now turned into something that might make a big national contribution. I'm quite excited about that.
Brilliant. Well, thank you so much for your time, Andrew. We will come back and talk to you later on when there's been more developments. And thank you to all the alumni who have supported this research.
Yeah, that's very kind of them. Thank you very much indeed.
Thank you very much.
Thanks. Bye-bye.